Very good. I'm glad. And sorry again for delay. Right. So I'm going to have this short presentation about quantitative genetics of protein glycosylation, and this is work we've been doing for a while with, mostly with colleagues presented at that seminar, like Gordon and Manfred were seminal in this work. I start with disclaimer. I'm full-time employee of GSK, and this presentation, however, is my personal opinion and is not that of GSK. I would like to start to emphasize or underline or trying to explain what is so special about genetics as a tool to study human biology, quantitative genetics in this case. And the thing is that, well, we are biologists here, and well, some of us are medical doctors, so we all know very well that humans are, well, very difficult object to study, right? While when you study other species, you have a lot of space for experiment and to establish causality in biology. When you work with humans, you are very limited in your experimentation. And in fact, when you want to establish causality, there are only two things in the arsenal of a biologist. And one thing is randomized controlled trial. The thing is that this is a very expensive thing to do, firstly. And secondly, not every experimental modification you can think of would be liable to this technique. The second possibility to establish causality is, in fact, genetics. So what genetics does is following up on the experiments of nature. So basically, what you do, you track the effects of random mutations which happen at random all the time, right? So you can see how disturbance of specific genes translates into observed phenotypic variation. Now, it should be mentioned that the applicability of genetics as a tool to study biology has very clear scope, right? So here we can think of the core biology and if you wish, peripheral, or maybe better word is variable biology, right? So when I talk about core biology, these are the things which you see very little variability in human population, right? So if you think like the number of hearts we have, the number of kidneys we are born with, number of hands, etc., right? So this all is core biology. And then, of course, any mutation which really disturbs genes encoding core biology, in fact, you are not likely to see the effects of this very genetic variation, of this mutation on phenotypic variation, because these people don't live, right? You don't see them in the population, and moreover, these genetic variants which lead to the disturbance to core biology, they are not propagated, they are not transmitted in further variation, right? So genetics, germline genetics, is not a good tool to study your core biology because you don't see variation for these traits for the core biology in your population. However, as you go more and more to the traits which you see variation in population, the more is potential of genetics and specifically quantitative genetics to study these variations, right? We see a lot of variation in things like height of the people, levels of different metabolites in blood, propensity, susceptibility to different diseases, right? And all this is part of the variable biology. And when a trait is a part of variable biology, it is liable to the study of causality through the genetics approach, right? We can see which genetic variants lead to the variation in the trait of interest. We can track this variation down by measuring genomes of people and connecting the variation in the phenotype to the variation in the genome, and tracking down the biology of this variation to specific genes. Okay, so title of my talk is specifically quantitative genetics of human protein N-Glycosylation. And quantitative genetics deals with the quantitative traits which are variable in general human population. So this is this variable biology. And the approach we use a lot, and specifically which we use to study the populational variation in protein and glycosylation, is genome-wide association study. This is the approach where you measure the genomes of thousands of people, and on the top of the genome, you put your trait of interest, and then you relate the variation in the genome to the variation of the trait of interest. In our case, this trait of interest is protein and glycosylation, and I will tell you a few more words after that. So after having this introduction into quantitative genetics, what are the questions we are specifically interested in to study with the genetic tools, right? So we all know that in principle, the biochemistry of protein and glycosylation is very well studied, right? We know which enzymes, which glycosyl transferases work in which specific cellular compartments to add this or that glycan on the building blocks, right? Now, then, like the question would be, why would you need this very special, very strong, but sophisticated tool as quantitative genetics, which specifically allows you to study in vivo human biology and establish causality and biological links, right? So the thing which we actually don't know are that what are the tissue-specific networks of gene that regulate protein glycosylation. So this, in terms of both humans and non-humans, is studied much less, right? And then the second question, which we were thinking we would address with quantitative genetics when we started this story about 15 years ago, was we've seen observational studies, and this is what Gordon partly presented, that there is association, epidemiological observational association from observational studies between different abundance of different glycan structures and the disease. And some of these things were established in follow-up studies. Some of the things are seen in cross-sectionals. And then it's often is a little bit difficult to understand what is the cause and what is consequence. So the question of causality, but secondly, what is the mechanism underlying these associations? And here where genetics can also help, because it breaks down, disentangles, it allows you to disentangle complex biology into the building blocks of nature, into the genes, right? So you track down your variation, phenotypic variation, to the effector genes and gene codes for a protein. So this is like minimal unit of biological function, right? So again, like what is the nature of association between protein glycosylation and complex diseases? This is interesting and difficult question. And this is the one where quantitative genetics can be very, or genetics in general, can be a very effective tool, right? A lot of work was done over previous 12 or 15 years with a lot of impact from Gordon, Manfred, and other colleagues present at this seminar. We summarized multiple genome-wide association scans and the results and biology which we learned from it in this paper published in Engineering. So what I'm going to do in the next few minutes to summarize the results which we report in details in this publication. So if you're interested in more details, I'm referring you to that paper published in the special issue of Engineering. Very good. So what we were able to study using quantitative genetics approach in terms of human protein and glycosylation. So the one thing is that you need big sample sizes, measured both genomes and their N-glycomes for this approach to work, right? So we are talking about minimally thousands of people, right? So this is high throughput, this is expensive, therefore you cannot often go very deep, right? So the three domains of N-glycosylation we've been able to look in effectively is glycosylation of specifically immunoglobulin G, of transferrin, and the plasma glycol. So if I use the analogy of Gordon, which he just offered us in his first talk, right? So he had this analogy with the parrot, which has feathers or have no feathers, and then these feathers are basically glycans of the protein, right? So when we study IgG and glycosylation, we are looking at feathers from one bird, and this bird is IgG. When we looked into the transferrin and glycosylation, this is another bird, and we were looking into feathers of this bird. But when we are looking into plasma glycol, we are looking into a bag of feathers, which we have from many, many proteins, which are glycoproteins from the plasma. Okay, so what have we learned from studies of N-glycosylation of the two proteins which are prevalent in human blood plasma and total plasma and proteome? So we do see that variation, and here is important detail, right? So these are the candidate genes, and in most of them we have high degree of confidence, but it's not always 100%, and this is due to the specific of the method, right? Because this mapping doesn't allow you immediately and always map the phenotype of interest to exact one gene, right? Sometimes you have a doubt whether other gene in the region may be. So with this drawback, this is our best take on the causal genes in the 34 loci, genetic variation in which was established to be associated with changes in human protein glycosylation, right? And I mentioned that we've been studying three domains, and the one most studied is N-glycosylation of immunoglobulin G, and there have been 5 studies published, and the sample sizes were up to 8,000 people. So quite a few genes were implicated from this work. The second domain is N-glycosylation of transferrin, and then this is only one study published quite recently by Gordon and us. About 2,000 people, and these are the genes implicated through transferrin studies. So you can notice that this bunch of genes is very specific for transferrin and glycosylation, while these three do overlap between IgG and glycosylation and transferrin and glycosylation. This is an interesting observation. And finally, this is the group of genes which were implicated in studies of total blood plasma and glycosylation, 3 studies with up to 3,500 people, right? And so this is the result based on which you can build your further understanding, right? Because these genes are telling you a lot, and you can also look into a lot of data generated about these genes in the field of functional genomics and computational biology for other purposes. So this gives you the starting point of very deep dive into N-glycosylation biology, right? One thing which you can see immediately that in blood plasma, you don't have any gene which is specific for blood plasma only. It's always either coming from, you can see that it's also affecting variation of N-glycosylation of transferrin or IgG or both. So one of the first conclusion is, and this is kind of natural, but it's nice to see this to be recapitulated on this level, is that plasma in glaucoma is driven by glycosylation in the lymphoid and hepatic tissues, right? So we can see that transferrin as a representative of liver secreted proteins, while IgG is definitely produced in antigen processor cells, right? So you think of glycosylation machinery in these two tissues working quite differently, but basically the machinery of these two tissues is defining for what you see in plasma and glaucoma. The second observation is that, so when we look in this gene, we see the specific biological mechanism affecting N-Glycosylation are recapitulated. They are all known, but that we see it on this level, on the level of genetic variation, tells that this specific mechanism can be studied further in terms of deep biology, high confidence biology, and causality using the quantitative genetics approach. So we see glycosyltransferases, we see transcription of factors which regulate these glycosyltransferases. We see proteins involved in Golgi pH, in glycoprotein transport and degradation. And we also see that variation in some glycoproteins, the abundant ones, do change protein glycosylation. This is a very good example of structural variation in transferrin affecting its glycosylation. So next, genetics recapitulates biological overlap and in fact starts offering the clues to the mechanisms common between glycosylation and, as Gordon mentioned, we see a lot of overlap also on genetic levels with determinants of risk to cancer, cardiovascular, autoimmune, and inflammatory disease, these classes of diseases, right? What we see on also big picture is that the genes involved into regulation of glycosylation in antigen-producing cells, they are overlapping with autoimmunity, inflammatory disease determinants, while what we see for liver is more towards cardiovascular disease and its risk factors. Then next observation, which is rather fundamental, is quite interesting, that what we see is that the expression in terms of RNA expression, for example, of many glycosyltransferases and regulators of glycosyltransferases are tissue specific, right? But genetic determinants seems to be even more tissue specific than the expression level regulation, right? So even when we see that specific glycosyltransferases like FAT8 or FAT6, they are expressed both in liver and in antigen-producing cells, in B cells, right? Then still the genetic regulation, like the variation which will affect the expression of these genes in different tissues, will be different. So we think it's something that enhancers, a tissue enhancer specific variation, but we need to prove that. Okay, so this is like last slide, and this is more a speculation. A picture, very interesting picture, which appears is that it seems that at least in N-Glycosylation, and this is quite unique, I've been mentioning this core biology and this variable biology, and it seems that at least in glycosylation, there is this correlation, temporal spatial correlation between core biology and peripheral biology, and the process how glycans are made. So we don't see much variation in this core process where the glycan chains start in building up, right? And this makes a lot of sense, right? And the populational variation affecting the observed variation of glycans, this is mostly concentrated at the terminal stages. And I find this, as a geneticist, I find this observation very interesting. Right. With that, I finish my presentation, and thank you very much for your attention.
工程创造人类美好未来。